I'm Matthew Ryan. I'm Research Leader Biological Resources at CABI in the UK, an international organisation, and also the PI of a new project called the UK Crop Microbiome Cryobank. In this presentation, I'm going to provide an overview of the current status of culture collections throughout the world in order to provide an understanding of what collections and biobanks need to do to underpin the microbiome research community. I'm then going to talk a little bit about the EU Microbiome Support Project and our new UK Crop Microbiome Cryobank, and finish off with a slide on cryopreservation research, which has been the focus for much of my career. To give you an indication of what CABI is, if you've never heard of us before, we're a not-for-profit international organisation that improves people's lives by providing information and applying scientific expertise to solve problems in agriculture and the environment. We're a member country-based organisation. We have 50 member countries, our newest being Ethiopia. We also have a strong presence in South America. We have a Chile as a member country, and we also have offices in Brazil and activities elsewhere. I have been a collection curator for much of my career, and it's been an honour to curate a collection of 30,000 living fungi and bacteria. Perhaps our most famous strain is Fleming's mold, and it was only recently that we managed to do the full genome sequence of this particular strain. And being that it's a Fleming mold, it got quite a bit of global attention, including this article on CNN. So firstly, what's in a definition? What is a culture collection? What is a biobank? How do they differ? These are quite contentious so it's only a loose grouping. A culture collection is typically a resource of living exenic microorganisms, but it can also extend to animal cells, stem cells, algae, and the associated data, whereas biobanks tend to be more medically orientated, but of course not exclusively. Then we have the added definition of the biological resource centre, or the biological resource collection, which is defined by the OECD to underpin biotechnology. Collections may also be known as cryobanks, genetic resources collection, microbial resource collections, and a whole gambit of other names as well. So the terminology is quite important. But what is the role of a traditional collection? And it's important to understand what the role of a traditional collection is if we are to extend it to cover microbiome. It's typically the preservation and supply of biological resources, be it cultures or DNA. We underpin research and development, and to a lesser extent conserve biodiversity. That it really is fundamental to support the science that we do. We also have to operate to the Nagoya Protocol, for example, and also international standards, all things that we need to develop for microbiome. Of the big culture collections throughout the world, these are the fungal ones. ATC in the States and the Westerdyke Institute are the best, but there are many others dotted all throughout the world. These are the fungal ones, but of course DSMZ in Germany is another major microbial collection. Last year, 2 years ago now, we reviewed the status of fungal biological resource collections throughout the world, and we looked at these from an international development perspective. We found that there's 3.2 million strains available throughout the world. So this is before we look at microbiomes. These are exenic strains. But when you drill down on these figures, 75% of them, the currently described fungi, are not available for living study. That basically relates to the fact that fungal diversity is represented rather poorly in collections. It's a similar story with bacteria as well. But for us, the most shocking thing is when we looked at where culture collections were throughout the world, the majority are in North America and Europe. One of the most diverse regions of the world, Africa, has only 17 culture collections as it stands at the moment. So that's one culture collection for every 75 million people, compared in Europe to one culture collection for every 3 million people. But these are exenic cultures. And as my colleague, the eminent mycologist David Minter said, if we're unable to conserve the fungi, the vital components of any ecosystem, then what good are the seed banks and zoos in the event of the Armageddon? So seed banks are fine, but if we're going to use them for ecological sort of repurposing of the environment, we need to maintain the fungi as well. And that's where the challenge lies for us in the microbiome community as well. How do we underpin this really important research field? The EU Microbiome Support Project has tried to look at this across the domains. And this is a brilliant infographic of how everything interacts, from the human side of things right through to the agri-food. And it's really important that we approach this as a community, not just the medical community, not just the agricultural and environmental communities, but we do this together. Another really useful initiative is the International Alliance for Phytobiomes Research, which really emphasizes the importance of microbiome in the higher phytobiome system. This is establish a science and technology foundation for site-specific phytobiome-based enhancement of sustainable food, feed, and fiber production. And biobanking of microbes and microbiome samples is an important factor if we're to achieve this. The EU Microbiome Support Project, we got a group of scientists from around the world together to look at how biobanks can support the microbiome community. We know that the current biobanking infrastructure is fragmented and it's not prepared for the rapid advancement of microbiome biobanking. And our main conclusion was we need to have an urgent assessment of how biobanks and culture collections can underpin this particular field. And this slide really emphasizes that point. We have the culture collection community on one side and the biobanking community in another side. There's very little interaction between the two. Then we've got the major data and bioinformatics initiatives, such as the Global Genome Biodiversity Network, the WDCM. And then underneath that, we have the various EU infrastructures and projects. So there really is a mechanism and a need to get these together to support microbiome research. Then we looked at the question, well, what actually do we need to keep? We know microbiomes are dynamic. They can be synthetic, natural, they can be constructed or manipulated. Should we store them dead or alive? And actually, where do we start? Do we look at preserving microbial consortia, whole microbiomes, exenic microbes isolated from those? We have to consider the data. So there's so many things that we need to bring together to look at and to store. But in storing the microbiome, we have to take a step back. And Douglas Adams in 2002 said, if you try and take apart a cat to see how it works, the first things you have on your hands is a non-working cat. And my colleague Tom Bell at Imperial College nicely framed this when he said, discovering which part of the community can be taken away without altering the functioning of that community structure are really important. How do you differentiate between a heart and a whisker? How do we know what is important in those systems and what is not important in those systems? And if we're to cryopreserve these samples, what are we actually doing to that structure of those communities when we cryopreserve it? We've started to look at that now with the UK Crop Microbiome Cryobank. Our aim is to establish a cryopreserved and characterised crop microbiome resource in the UK, and we got funding to start this project in November. The aim is to facilitate a comprehensive platform to facilitate research towards optimising plant yield in an integrated crop management environment, and Kew Gardens are also involved in this project as well. We'll be looking at major crops, but most importantly, we'll be using cryopreservation technology to conserve the components of the functional microbiome, while also doing full bioinformatic analysis of the samples that we will hold by cryopreservation in the cryobank. And this perhaps brings me on to the most important aspect of what I'm talking about this morning, and that is the very cryopreservation research that we need to use to underpin our activities. Ultra low temperature of sample is the optimal method of storage. We're talking at temperatures there of below minus 175 degrees. Why? Well, if we store warmer than minus 135 degrees, you can get recrystallization of ice. So storing things in a minus 80 freezer maybe is not a good idea. We must also preserve those organisms optimally. We should apply a control rate cooling approach to prevent the damaging effects of ice nucleation, but also apply cryoprotectants to help mitigate the damage to those samples. Once we've preserved something, we need to know how well we've done it. So we need to look at the effectiveness of preservation regime, which we need to evaluate using a range of genomic and more traditional microbiological methodology. And to underpin it all, we need to develop standards and quality systems to make sure that whoever does this in the world does it to the same standards, to ensure that we are really developing and producing a quality output. So, in summary, microbiomes continue to be a rapidly developing area of science. Culture collections underpin that very integrity and provide valuable resources for research activity. But collections themselves need to adapt, and in association with biobanks and the microbiome research community, we really need to adapt and develop new horizons to cryopreserve these resources. And that this really needs to be done with a view of conservation of biodiversity as well. And finally, there's a need for everybody to work together. The medical, veterinary, food and agri-tech communities must come together. We can't afford to work in isolation if we're going to underpin this really important area of research into the future. Thank you. And I'd just like to acknowledge our research funders, that's UKRI, BBSRC and EU Horizon 2020. And once again, thank you for the organisers for inviting me to speak this afternoon. Thank you.